In this PPT lecture, we will be discuss about the lac operon. Firstly, we will start with the introduction, then the image which explain the overall mechanism of the lac operon, then the mechanism, some important points about the mechanism, then the reference which I refer. Let's start with the introduction. Lac operon are responsible for the breakdown and the transportation of the lactose. So lac operon is responsible for the breakdown and the transportation of the lactose. So lac means there is a lactose, and the lactose breakdown and the transportation should be takes place only when the lactose is present in the medium. If the lactose is present in the medium, the expression should be on. The expression should be on. When the lactose is not present in the medium, lactose should be absent, the expression should be off. So depending on the presence or absence of the lactose, the expression should be on or expression should be off. So lactose should be break down into glucose and galactose. So for the breakdown of the lactose into glucose and galactose, there is an enzyme is required which is known as the beta galactosidase. So beta galactosidase which are the enzyme which is responsible for the breakdown of the lactose into glucose and the galactose. So this whole breakdown should be takes place in the presence of the beta galactosidase. So after the breakdown, there should be release of the glucose and the galactose, but for the intake of the lactose inside the cell, there is the other one protein is involved, other one enzyme is involved, which is known as the permease is responsible to take lactose inside the cell, and some other enzymes are also involved. For example, transacetylase are involved to remove the toxic material. So this whole mechanism of the lactose breakdown and the transport was explained by the Jacobs and the Monod. So lac operon was explained by the Jacobs and the Monod, and both scientists gave the Nobel Prize about this discovery. And this is most important for understand the overall process of the lactose breakdown and the lactose transportation. Let's overview some points before starting the exact mechanism. So this is the enzymes, various enzymes which I explain and their role, which I these are the various enzymes which explain, that is the beta galactosidase. Beta galactosidase are the enzyme which are produced by the lac-Z gene, and the permease are the enzyme which are produced by the lac-Y gene. Transacetylase are the enzyme which produces by the lac-A gene. So beta galactosidase are the enzyme which produced by the lac-Z gene. So lac-Z gene produces beta galactosidase. Beta galactosidase is responsible for the breakdown of the lactose into glucose and the galactose. And the permease are the enzyme which present in the cell wall of the bacteria and which is responsible to take lactose inside the cell. So permease are produced by the lac-Y gene. Lac-Y gene produces a permease, and the transacetylase are the enzyme which is called as the thiogalactosidase transacetylase, which are produced by the lac-A gene. Lac-A gene produces transacetylase which remove the toxic galactosidase, because the toxic galactosidase are very toxic for the bacterial cell, and it's most important to remove such a toxic galactosidase, which removed by the transacetylase, thiogalactosidase transacetylase, which produces by the lac-A gene. So this is three main structural gene and gene enzymes which are produced by the structural genes, that is lac-A-Z produces beta galactosidase which break down lactose into glucose and galactose. Permease allow to the entered lactose inside the cell, and transacetylase remove toxic galactosidase. So suppose this is the bacterial cell which contains lots of lactose in their surrounding. This red color is the lactose in their surrounding, and if the lactose is present in the surrounding, bacteria take lactose inside the cell by using the permease. Permease are present into the cell wall of the bacteria and which allow lactose to enter inside the cell. Whenever lactose entered inside the cell, the mechanism should be on. When the lactose are not present in the medium, here the lactose is not present in the medium, no lactose in the surrounding of the bacterial cell, so there is a mechanism should be off. So depending on the lactose presence, the mechanism should be on. Depending on the lactose absence, the mechanism should be off. So suppose this is the medium, this is the luria broth, This one is also the lurea broth, and both lurea broth contains the E. coli. This is E. coli cell. This is E. coli cell, and in the first media, this one media, in this media, it contains the lactose, and it does not contains the lactose. If it contains the lactose, then bacterial cell should be take a lactose, and mechanism should be on. If there is no lactose, the mechanism should be off. So depending on the presence or absence of the lactose, the mechanism should be on or the off. So beta galactosidase produces the beta galactosidase break down the lactose into glucose and galactose, and beta galactosidase produces by the laxate gene permeates allowed to enter lactose inside the cell, and thiogalactosidase remove toxic galactosidase from out of the cell. So this is overall mechanism. This is overview lecture. In this next lecture I will be explain about the how the mechanism should be occur.